So as I mentioned, we make these stimuli responsive biomaterials. We can make them tunable or cell responsive. We can also tune their properties. So these are injectable materials. Think of these, for example, liquid bandage for wound healing applications. For example, we can apply these hydrogels to improve wound healing processes. And we use these biomaterials to understand how cells interact with these biomaterials and how we can then control the properties of cells by changing or tuning the properties of biomaterials. Another key theme in my lab is that we apply these biomaterials basically to solve problems. The key thrust area or thrust of my lab is to develop these biomaterials that we can use to grow tissues. For example, in an NIH-funded project in my lab, we are using biomaterials to repair damaged cornea. In a recent project, we are using these biomaterials as injectable matrices to repair brain damage. So when the stroke basically damages the brain tissue, the idea is that can we inject biomaterials loaded with different drugs to improve the regeneration processes in the brain, which is a pretty hard problem to do. Another key focus in my lab is developing a whole organ for transplantation in patients who are in need of organ transplantation. We are pretty much focusing on liver in this case, where we are using bioprinting technologies together with biomaterials, looking at cell matrix interactions, using stem cells, and combining all these technologies together to see if we can create a liver in the lab that can be transplanted in human patients to sustain life in them. This is the overarching goal of the lab. Okay. So what I'll do in this presentation is I'll just share a couple of stories from my PhD research and then the recent research from my lab in developing novel biomaterials. And I hope that you'll be able to understand how we are leveraging our approaches to basically solve these problems. Now, the first story relates with a disease called corneal endothelial dystrophy. And again, as I mentioned that, I understand this audience may not be very much familiar with healthcare research, so I'll try to make it easy and less dense. So there is this disease called corneal endothelial dystrophy. Cornea is basically this front transparent portion of the eye. And so when we see this cornea, the innermost, it's a layered structure. The innermost layer of this cornea has this single sheet of cells called endothelium. But due to diseases such as endothelial dystrophy, these cells start to die. And so when this cell layer is gone, the cornea becomes cloudy. And you might have seen patients with this kind of cornea or cloudy cornea. There could be other reasons, but almost 40% of corneal transplants are performed just because this layer is damaged. This single sheet of cells is damaged, and these cells cannot grow again. And this damage happens because on the posterior side of the cornea, these bumps or microstructures start to appear that eventually cause the cell death. So how do we fix this issue? Currently, there is no medicine to fix this issue. So what we thought during my PhD, so I was collaborating with Dr. Jawad Mehta in Singapore Eye Research Institute. So what we thought that instead of doing a normal cornea transplantation into a patient, which is only good for treating one patient, why don't we isolate these cells from that donor cornea and try to grow these cells in the lab so that we can treat multiple patients? Because we can grow the cells in the lab, so which means that if we start, let's say, with 100 cells, we can maybe get 2,000 cells. So we can potentially treat more patients by either doing cell injection therapy or by transplanting a sheet grown on a hydrogel film into the patient eye. So we did a lot of work revolving around these techniques and improving different aspects of these approaches during my PhD, and we are continuing towards basically refining these approaches for clinical translation. So what we first did is the key challenge in this is that the cells do not really like to grow. These cornea cells, when you take them out of the cornea, they just don't like the environment. You put it in a dish to grow these cells. they hate it. For example, you see that the passage at different passages or different days during cell culture, we use this image analysis technique to basically show that the cells start to gradually become bigger. So the cell area increases over different passages, the cell density decreases, and the cell circularity also decreases, which indicates that the cells are kind of losing their characteristic shape, which is a marker that these cells are differentiating into other cell types. Basically, these are not the pure corneal cells when you try to grow them in the lab. For example, in this picture, it shows that some areas of the cells, they are very nicely expressing the proteins markers that they are supposed to express, while in certain regions, they lose those markers. So again, indicating that these cells kind of lose their markers. Now, clinical trials, for example, in Japan have showed that if you inject these immature cells, they don't really work well in human patients. So the cell injection therapy is pretty inferior unless you have ways to grow these cells in a better conditions. So we started looking into, because this is all published, so I'll quickly go over this work, that we thought that when we grow the cells in the lab, one thing that's missing is the surface topography or the surface patterns. For example, in vivo, the corneal cells are present on these micro and nano sort of structures. But in the lab, when we grow in petri dish, those structures are missing. So we thought that can we recreate some of those structures and then start to see how we can control the cells by using biomaterials with these pattern surfaces. And indeed, when we compared cells which were grown on a flat surface compared to cells which were grown on different type of topography, such as pillars or wells, we could see that in certain configurations of pillars, the cells started to express those markers which were missing when they were cultured on an unpatterned surface. Well, this is simply showing that the cells are able to recognize the surface and they are able to grow better on certain type of patterns, or as we call them, topographies. Then we started looking into the effect of different cases where if the cells are grown on topography versus if they're grown on a flat surface, if we passage them, are they still able to maintain their functions? And we did see that if you grow them in topographies and then passage them again, they're still able to maintain their functions, which means that the cells grown on these topographies are better cells for cell therapy applications because they still maintain the improvement in their functions even after they have been passaged. And this was pretty much actually the motivation of this was provided by a paper published in that around 2014, 2015, that showed that the cells can remember their mechanical environment. And that mechanical environment influence their fate. So we designed that experiment basically to see if the corneal cells can also remember their environment. And indeed, they do remember. In my lab now, we have actually gone further and we have started looking at cells which are freshly isolated, and we compare those cells with the cells that are cultured on plastic for almost 28 days. Now remember, plastic is very hard surface compared to a human cornea. And by now we know that the mechanical signals or mechanical environment strongly influences cell functions. So we did RNA sequencing. This is an approach where we can basically analyze all the proteins expressed, all the RNAs basically that are expressed in the cells. And we performed the PCA analysis or principal component analysis, which showed that the culture cells are indeed very different compared to the native uncultured cells, possibly because they have been cultured on plastic, which is an unnatural environment. We went ahead and did a bunch of analysis on gene expression and showed that almost 276 genes have over 5-fold change in their expression. And a lot of these genes are implicated in mechanotransduction. Again, I'll keep it simple. I'll skip a lot of details here. But what we are really trying to do here is we're trying to use more advanced techniques to see if we can identify which signaling pathways or which mechanisms are involved through which cells are sensing the environment, so that if we have better knowledge or better understanding of these mechanisms, we can design better biomaterials that can maintain these cells in a growing phase without losing their functions. With an overarching goal is that we want to produce better cells for cell therapy applications. So in my lab, one of the key projects that we are doing is we take donor human corneas, we isolate these human corneal endothelial cells that we are then growing in plastic and also on hydrogels that can mimic the biomechanical properties of the human cornea. And the goal is to then analyze the gene and protein expression analysis and then further identify the mechanotransduction pathways that can improve the expansion of these cells. Okay. Now, another approach, I mentioned that this all happens due to a disease which causes these microstructures on the cornea. So we thought that can we use microfabrication technology to sort of mimic the disease environment in the lab? So for example, can we make these lens-like structures or these pillar-like structures and grow cells on these structures and see what happens? Can we mimic, or in other words, can we make an in vitro disease model? Can we recreate the disease in the dish so that we can perform some disease modeling or investigate what really happens to cells when they grow in these conditions? And indeed, I'm going to skip again this slide. What we see that if the cells are growing on this flat surface, they're able to sort of migrate easily and they can maintain their monolayer shape. But if they are grown on these pillar-like structures, the cells are very elongated. So if you notice that the cells are more elongated, if I can play again. So if you see that the cells are more polygonal in shape in this case, and they are fully covering the surface. But in this case, it's the same time period, 24-hour video, the cells are more elongated. And even after 24 hours, they have not fully covered all the field of view here. So it means that these structures really obstruct or inhibit the migration of these cells. So this is the first time we were able to recreate the disease environment in the dish that showed us that the cell therapy in those patients in which the cornea is also already in this kind of state may not work. We first need to remove these damaged or these diseased cornea surface before we can inject cells to treat patients. And our observations really mimic the patient samples. For example, if you look at patient samples, we know that gottata or that microstructure contains a lot of nearby cells, and we were able to mimic the same conditions basically in the lab also, where if you see that that pillar contains a lot of cells that are surrounding this the pillar structure. Now, another approach we can use to basically treat patients by using these cells is that we can grow a monolayer on a hydrogel film, and then we can transplant that film into the cornea. Now, I started using gelatin methacrylate as a hydrogel for cell carrier. That has basically excellent cell adhesion. It's highly tunable hydrogel, so we can tune the mechanical properties, and it's also biodegradable. But it was mechanically a weak hydrogel. So corneal surgeon, they can't really transplant this hydrogel film because of the mechanical weakness. So we improved the cross-linking mechanism in these hydrogels, where instead of using the single cross-linking, we combine sort of physical cross-linking that happens in a gelatin solution with the UV cross-linking approaches so that we can make better hydrogel or more stronger hydrogel. And indeed, compressive modular analysis, for example, if you see these two groups, both have 30% concentration of polymer, but one of the hydrogel that contains this dual cross-linking is almost 5 times stronger compared to a hydrogel that has single UV cross-linking. So this approach we termed as dual or hybrid cross-linking with 2 types of cross-linking approaches that improved simply the mechanical strength of this hydrogel film. Then basically, because we knew that certain type of patterns can really improve cell functions, so we created those 1 micron pillar structures on this really soft hydrogel film, relatively soft compared to a silicon wafer, for example, or a quartz surface. So when we grew cells on these pattern Gelma Plus films, they were able to grow very nicely. They could express the protein that we were expecting them to express. This, for example, shows an SEM image of this polygonal cell sheet growing on a hydrogel film with patterns. We verified that the hydrogel was able to degrade in collagenase solution, so it was biodegradable. It could also degrade in rabbit aqueous humor. So aqueous humor is basically the liquid found in the interior chamber. So we collected aqueous humor with the help of collaborators and evaluated the hydrogel properties in that. And then we put that hydrogel in rabbit animal models. So here you can see basically this is rabbit cornea. This is the hydrogel film. Again, the goal of this hydrogel film is to transplant cells, right? The cell sheet. So the cells will be on this side of the hydrogel. So we evaluated the biocompatibility and biodegradation. So day 15, things look good. Day 120, which is 4 months after transplantation, things still look good, but the hydrogel film is still there. We initially thought that the hydrogel is biodegradable and therefore it will degrade. But it did not degrade. It was still intact in vivo even after 4 months. And that's a huge challenge. So let me explain why this is a challenge. Remember, our goal is to create a transplantable matrix on which we can grow a monolayer of cells or a sheet of cells. And when we transplant, this is the host cornea, this is the hydrogel film, and then on this side, we have the transplanted cell. If the hydrogel does not degrade, we can never evaluate how these transplanted cells will integrate with the host cornea. Therefore, we want that hydrogel film to be gone. Remember, the purpose of that hydrogel film is really to serve as a cell carrier and not be there permanently. But it did not degrade even after 4 months. Now, in biomaterial field or in hydrogel engineering, it's always a challenge. If remember, we wanted to also make this hydrogel strong so that it does not break during the process of transplantation. And the challenge is that if you try to make something strong, the biodegradation rate goes down. And if you want to increase biodegradation rate, the hydrogel strength will have to be lowered. How do we achieve high strength and also high biodegradation rate? So we thought that the regular engineering processes will not allow us to achieve this. So we'll have to be a bit more creative. And we thought that, is there a way to degrade this hydrogel by using, instead of relying on biodegradation, can we use some sort of stimuli to degrade this material? For example, one of the stimuli, we have a fascination with light. So we really want to use light to control the properties of biomaterials. So we thought I had previously worked with biomaterials that can be controlled by using light. For example, in this case, we used femtosecond laser or a 2-photon laser to show that this dimethoxynitrodibenzopyran group can be efficiently cleaved to basically expose reactive thiols, and we then conjugated some dye with it and showed that in 3D hydrogel volume, we can nicely control the properties. So, and the same compound can also be used as a crosslinker to sort of crosslink the gel or to hold the gel molecules together. And when we shine light, the idea was that maybe we can degrade it. What in my lab we are currently doing is we are developing these cleavable crosslinkers, basically crosslinkers that we can use to crosslink or convert liquid hydrogel to a solid hydrogel. And if these crosslinkers are responsive to near-infrared light, then we can shine near-infrared light to degrade this hydrogel when needed. Another version of a biomaterial we are making is responsive to sortase, which is an enzyme and can be used as an eye drop. With these approaches, the goal is that we can transplant the hydrogel and then later either shine NIR light or expose the eye to sortase enzyme. This way we can degrade this material, and then this will finally allow us to evaluate the integration of the transplanted cells with the host cornea. Because this is a problem no one has been able to figure out so far, that how do we evaluate whether the transplanted cells will actually integrate with the host cornea, because biomaterial is always in the way. And going beyond simply this project, these biomaterials serve as a platform because they are stimuli responsive, so they can be used for stimuli triggered drug release, or harvesting cells after 3D culture inside these matrices. So we are pretty actually excited about these biomaterials because they